Plenary session is ended and we can start our section. Welcome all the participants, the lecturers and audience in annual Wellman conference in food processing and safety and rural and sustainable development session. First of all, I would like to introduce our organization team of this session. First of all, I can introduce the co-president is Adrian Sarvas, and I am the president of this session, Anita Jiba. We are glad to organize our conference in this year with 55 participants, and we hope that the next year comes more and more participants year to year. I wish you a pleasant and useful staying at the Vermont conference, and I would like to inform about the details of the participation. We would like to ask the maximal time of the lectures, 15 minutes, and the short lectures, 5 minutes. The uploaded presentation we will upload before your lectures. But now
Sorry about, I think there is some technical problem. We wait until Anita is coming back. Until Anita join us back, I also would like to thank everybody that joined to this conference, and we hope we will listen a very good scientific lectures today. And yes, I also want to, because Anita was saying that you watch very important the time, because we are already start later. And end of the, when you finish your presentation, do not hesitate to ask questions to each other. You are welcome to ask any question. So I also want to say everybody thank you. And I just call Anita if maybe we can start, because now it's so late. Just 2 minutes. Okay, we will wait 1 more minute. And I just called Anita. She's coming back. I think it was problem that because we put our web camera on. So I mentioned for her maybe it's better if we are not using the camera.
sorry, it was a technical problem, but we start with the first uh, lecture titled Textural Investigation of a Protein-Rich Jelly Sweet. The lecturer is Adrian Varga Tóth. I ask Adrian Varga Tóth to start your presentation, please.
Okay, so welcome everyone. I would like to ask you to give me a feedback if you can hear me.
Yes, it's very good. We can hear you.
Oh, okay, thank you. I thought already I am lost in the internet. So if you don't mind, I won't turn on my camera because I think I may have some internet connection issues as well. So thank you for everyone. My name is Adrian Varga Tóth, as you mentioned. And today I would like to give you a short presentation about the textural properties of a novel product or a new product development. So I think that we can already start. So let's go further. So what about food development? I think that the basic point of food development is the consumer point of view, or to figure out what's the preference of end users. So for instance, we can see that in these days, food users or consumers are waiting for products which are having a kind of extra protein source, like a higher protein content, or we are thinking about diet. For sure, we ladies do that, and that's why we try to figure out or follow a low carb diet on the other hand. Basically, we see that the consumers like sweets and all the sweet and confectionery products like chocolates, gummy bears, or other products that we can find in the shops prior Easter. But we realize that most of them are containing a lot of sugar, which might have a negative effect on our health. Conditions as well. For example, we will gain weight after or during the Easter season. So, in this study, we tried to investigate a new product that I will introduce you very shortly in a few minutes. So, jelly sweets. Jelly sweets are quite popular even in the international level and in Hungary as well. We call them even as gummy sweets or even chew sweets as well. Example, giving the Haribo babies or Haribo bears are maybe the most popular products in this product area. And these are familiar for everyone, I think, from the very, very tiny little childhood. So these sweets have a main ingredient called gelatin, basically, or we can use pectin, or we can use further things like carrageenan for providing to formulate, this kind of jelly texture. Basically, the best textural quality is given by gelatin. Gelatin is well known as a great protein source from different animal sources. And this is actually present in bones or in connective tissues or in skin tissues, for example. We can reach gelatin quite easily from animal byproducts as well, and during production of other meat or food products as well. Well, basically, we use gelatin in many, many kind of food products, especially in sweets, as I mentioned you. But during the Easter season, we see that in salty products we use it as well, like in different kind of aspic sausages we can use gelatin as well, or even in other products as well, called in Hungarian kocsonya as well. It's very popular during the spring season as well. So what about collagen? In nutritional point of view, it's a quite long protein chain that is quite difficult to digest. However, it is a great protein source. So collagen peptides are produced by several companies, providing several nutritional benefits for consumers. However, please note that these kind of collagen peptides do not have that gel forming properties like the initial gelatin, but they are easy to digest and they provide us a lot of protein and essential amino acids as well. So, just a moment, I have to change my slide. Okay. On the other hand, a great protein source for us, or an excellent protein source, is egg white, which contains a lot of high biological qualified proteins, such as egg albumin and ovotransferrin, for example. This is a great source in environmental point of view as well, because it's probably the cheapest livestock-based protein. It has anyway great technofunctional properties as well. For example, it creates a great foaming ability and capacity. For example, in sponge cake, we couldn't miss that technofunctional property, or in other foam-based foods as well. So, on the other hand, as I mentioned to you, these proteins present in egg white are great and very usable proteins for the human digestion. And today's consumer are searching for dairy and milk replacement products as well. And today we do have a kind of egg white based dairy replacement product called totu. The word comes from the word tojás-túró in Hungarian, and these products might be an excellent replacement or analogs for dairy products such as cottage cheese, yogurt, or sour cream, or mascarpone, or even as an alternative to cow milk. In this study, we used the cow milk alternative called totu beverage that I will show you later on. So the aim of this study was to work on, to start to develop a kind of product, a kind of jelly product that contains a lot of nutritious proteins, such as egg white proteins, as I mentioned to you, and gelatin, and even gelatin peptides as well. But in this point, I would like to tell you even that this kind of products we aimed to marketing for weight loss diet and during sports activities as well. So that's why we decided that we will use no added sugar, but we will create the flavoring by different fruit flavors, fruit purees actually. So what about the first samples? So the samples were created as jelly sweets. We used this totu beverage that was a milk alternative. We added erythritol for the sweet flavor. We used 3 different kinds of fruit purees, banana, red berries, and mango flavor as well. We used gelatin. As usually, you can use it at home. For example, after boiling the water, you have to add it in warm water, and after cooling, it will provide the jelly texture as well. And furthermore, we gave collagen peptides. These collagen peptides were bovine collagen peptides actually, and we tried to analyze its effect on textural attributes of these gummy sweets. We added the collagen peptides in 1, 2, or 3 mass ratio, and we created our samples in a muffin frame-like sample holder, and it meant that the diameter of the samples were 6.5 centimeter and its height was about 4 centimeter each. So after creating these samples and cooling these samples, we analyzed their texture attributes by using the stable microsystem or SMS system, and we figured out that the best method is the textural profile analysis. We chose this method after penetration and cutting methods as well, but we saw that this method might be the most accurate for measuring a jelly-like based product. For that purpose, from the previously prepared samples, we created a cylinder-shaped sample, which had a diameter of 12 millimeter and a height of 12 millimeter as well. For the method, we used a 70% compression. It means that the 12 millimeter height was compressed to 3.8 millimeters. So we really compressed the sample and we tried to analyze the textural changes. This TPA method shows what's happened with a bite of food during biting or during eating. For that purpose, we pressed these samples twice, and you can see on the right hand side that the first bite will give us a bigger or a larger energy or force. Then first of all we have to really press, and we have to really shape the whole texture of a sample or of a specific food. And then during the second part, we use the 70% compression as well. We compress again with till 3.8 millimeter the samples. And it's like we have a second bite on the same slice of food as well. So, what about our results? On the left-hand side, you can see the cohesivity or cohesiveness of the samples. So, it's evaluated under the first area of the previous slides given texture analysis diagram. Here you can see that there are differences between the fruit flavors as well, such as you can see that the banana flavored samples were more cohesiveness than the raspberry one. But the raspberry and mango flavored fruit samples or sweets had almost the same cohesivity. But the cohesivity is depending on the added protein ratio. I mean on the Covan collagen peptides ratio as well. Here you can see the higher ratio was added, the higher cohesivity was measured. Even when we analyze the springiness, we can see the similar trends as well. However, you can see that the differences are quite little. But between the samples, there were found a significant, statistical significant difference, but not between the protein ratios. And after that, calculating the chewiveness of the samples, which is actually giving really the force or the work that we have to carry out during chewing or during biting a slice of cake, slice of food, or the gummy bear, you can see that the protein enrichment has a significant effect on the chewiveness. The bigger or the higher protein ratio was added, the higher chewiveness was calculated by the previous results. You can say that even the raspberries and mango flavored samples had higher chewiveness than the banana flavored one. It's because the banana puree has a different texture than raspberries or mango puree. mango puree and red berries are actually almost the same textural fruit purees that we have used. As conclusion, I would like to say that, well, as I mentioned, the chewiness is statistically influenced by the protein addition, and even there are some differences between the different protein concentrations and flavorings as well. Further on, we would like to still develop this kind of products, and we would like to go on further investigation in textural and sensorial point of view as well. So as conclusion, however, we didn't carry out sensorial evaluation in this experiment. I would like to conclude that the fruit flavors have not just a textural impact, but real taste and smell impact on consumers. And they prefer the red berries because it provides a kind of acidity to the product. And this kind of acidity can a bit hide the sweetener by taste. So that's why I think that we will work on later on that red berry flavored samples as well in further studies. So I would like to say thank you for your kind attention. And if you have any questions, please let me know. I try to answer them. And once again, thank you for your attention.
Okay, thank you very much for your interesting lecture and interesting presentation. I ask the audience and another participant if you have any question for the lecturer.
I have one question for Adrienne. Because you mentioned that you use the banana, the red berry, and the mango flavor. And do you have a reason why you choose these three flavors? Why you didn't choose strawberry and others flavors?
Yes, for sure. So basically, we had previous pilot experiments, and for example, strawberry has a less acidic and less strong smell and flavor, if I can say it. So, so we tried to search for more specific flavors which are quite strong in themselves as well. Like, for example, a banana puree is a very, very sweet and very, very strong flavoring. On the other hand, previously we concluded that red berries, which are containing strawberries, blueberries, raspberries, and so on, are great because they are quite acidic. And in such products, they are having a great smell and taste forming ability. I mean that kind of acidity you can find even in the gummy bears as well, and depending on their initial smell or flavor. And with mango, we had the same as well.
Mm-hmm. Mm-hmm. Thank you very much. Yes, I was so curious that what was the reason. That thank you very much your answer.
Thank you very much.
Okay, thank you very much. Have you any questions? Okay, if you don't have any questions, we continue with the next lecture titled The Effect of Different Temperatures on the Liquid Extraction from Pleurotus ostreatus, in French, cum mushroom. The lecturer is Greta Törös, and the co-authors, Dr. Ferenc Peles and Dr. József Prókis. I ask the lecturer to take your presentation.
Sorry, Anita. I tried to upload, but it's not letting me to upload the PPT. Just a
I
minute.
ask the lecturer, please try to upload your presentation.
Okay, thank you, because okay, I think I can now. I can
It's a
now upload.
technical break.
Okay.
Try
Now I
to
can.
upload the presentation.
1 minute.
Okay.
Okay.
Okay, thank you for the uploading. I ask the lecturer to start it, please.
Welcome to everybody. My name is Greta Törös. I am a first year PhD student at the Doctoral School of Animal Husbandry, University of Debrecen. During my presentation, I want to talk about the effect of different temperatures on the liquid extraction from Pleurotus ostratus mushroom. Elnézést, hogy tudom tovább vinni a következő.
Anita, át tudod adni a jogot Grétának?
Elvileg átadtam.
Átadtad?
Most
Aha.
Thank you,
tudod,
I
most már
see.
nincs. Rákattintottam, most kerül? Oké.
Okay.
Okay.
Thank you.
So the studied mushroom attracted several researchers for more investigation about this mushroom and its different application. The oyster mushroom has great nutritional and medicinal attributes, which make this mushroom to be the second most popular edible and cultivated mushroom in the world after Agaricus bisporus. Among all the several health benefits reported by many numerous published studies, this mushroom has also shown promising potential for green biotechnology, biorefinery, and recycling of agrowaste, as well as pharmacological potential and medicinal properties. In recent years, there has been a growing interest in extracts and isolated compounds present in the oyster mushroom due to their potent antibacterial, antifungal, antiviral, anti-helminthing, and pharmacological effect against different microorganisms. Furthermore, in the case of the potential growth stimulation activity of probiotic bacteria. The literature data indicate that the oyster mushroom contains a high range of phytochemicals with antimicrobial and probiotic function, like alkaloids, steroids, flavonoids, saponins, tannins, phenolic components, glycosids, and terpenoids. In recent years, the effect of cooking methods on mushroom quality has just been a little bit explored. Researchers mainly focus on higher temperature heat treatments, so further studies are necessary to explore lower temperature cooking methods. The low temperature long time technology, like sous vide technology, has advantages like less chance of forming toxic chemicals, for example, acrylamide, and lower vitamin and mineral degradation. However, more studies are necessary to perform. It is worth to mentioning that during this procedure, the taste and smell can be also improved. The shelf life of the product can be lengthened. During low temperature, long time cooking, Maillard reaction product can be formed, which has great nutritional and medicinal attributes, for example, antioxidant, antimicrobial, prebiotic, and antihypertensive uh, activities. The highest focus is on their valuable antioxidant potential. The topic actuality next to the gentle cooking process is that the extraction of the mushroom liquid without adding solvent is a significant technological, is more economical and is suitable to dissolve water-soluble bioactive compounds, for example, beta-glucan. The pleuron is one of the most frequently used beta-glucan with antimicrobial and prebiotic function, which can be linked into the protein. The significant focus is on beta-glucan's bioactive component due to their potential antimicrobial and prebiotic function. All in all the mushroom extract produced by the usage of the high amount of 70 and 95% of water content of the mushroom is expected to be rich in aromas and bioactive components. So, the main aim of the present study was to develop a method for the extraction of mushroom liquid without added solvents. So we divided the experiments into three parts. The first one was in the case of the manual filtering, the second was the centrifugation, and the last one is the modeling of the speed of bioactive rich mushroom liquid flow. We mainly focus into the Soviet technology, which is a water-based cooking method and which raw food material is cooked inside vacuum pouches under a precisely controlled environment. Fresh first quality of oyster mushroom were collected from Magyar Gombakertész Ltd. Demje. First, the heat treatment of cleaned and sliced oyster mushroom in vacuum packages was carried out in the drying cabinet 60, 70, and 90 Celsius next to 24, 48 hours, and 1 week. The manual filtering was tested and the amount of extract and production loss was calculated in percent. After that, the vacuum packages. Furthermore, we changed the manual filtering into 1004 RPM centrifugation next to 90 Celsius and 24-hour heat treatment. Dairy industrial filter papers used to avoid the clogging of the centrifuge and assure proper filtering. Finally, the average amount of mushroom liquid and dry water was identified. Arduino, an open hardware software used to study the mushroom liquid flowing across silicon tubes during 80, 85, and 90 Celsius and 15 hours. Silicon tube attached to an autoclavable bottle with 150 grams of sliced mushroom. The bottle was in a drying cabinet in kept-down position. The tubes are placed into the Meyer flux, which is on a balance. The balance is connected with Arduino software. The liquid flowing was monitored continuously in every 30 seconds by balance. As you can see in this first figure, in the case of the 90 Celsius 1 week and manual filtration, the percent of the extracts was absolutely zero. Furthermore, the average production loss was extremely high. The most affected liquid extraction, 33.6%, was at 60 Celsius and 48 hours. The lowest production loss has happened at 70 Celsius and 1 day. In the second series of the experiment, the extraction process was successfully improved. You can see in this figure that 57% of mushroom liquid can be extracted with centrifugation from the first quality of oyster fruiting body. However, 5% of production loss can be expected, which should be decreased in the future, for example, involving new manufacturing steps like fast cooling before centrifugation. As you can see in this figure, there were significant differences between the tested procedures. The liquid extraction was higher with 23% than the highest amount of the first experiment. Furthermore, we could reduce of the production loss with 19.03% with the usage of centrifugation. According to the result of the weight monitoring test obtained by Arduino software, without physical impact, 2 hours is enough for the fluid flow. The necessary tools have been built and developed. It was shown to be a proper tool for the quality control process of cooked mushroom. The next step is to involve a cure thermometer into the sample holder and test of the other mushroom species like Agaricus bispolus and Lentinula edodes shiitake mushroom. So, for the conclusion, based on the results, it can be said that the manual filtering is not an option for the identification of mushroom liquid extraction due to the huge amount of evaporation caused by the speed of the processing person. The usage of a pressure cooker and centrifuge can be also an economical method for industrial production. The quality of mushroom is mainly affected by the applied cooking methods. The usage of Arduino software and the build system has shown to be a proper tool. So the future research should include a more comprehensive study of the development of a ready-to-use system to analyze the changes, for example, the speed of the heating of the raw matter, cure temperature. So a complete monitoring system should be developed to study the changes in the physical attributes. Furthermore, the water-soluble ingredients should be also analyzed in the final products. New functional products should be also developed with the usage of the mushroom liquid. Like any scientific theme, more and further open questions concerning the mushroom of Pleurotus ostratus and its handling should be also answered. Thank you very much for your attention.
Okay, thank you for your lecture. Your topic was very interesting and important topic. First of all, I would like to ask, why did you choose this topic?
Because I found this mushroom very economical and environmental friendly, because for the oyster cultivation you can use byproducts, and it can be also easily growing, and I am planning also to learn how to producing this mushroom, and after that I would like to use for my next use the samples for my next experiments.
Okay,
And
thank you. Please.
and also it's very strong in fiber content. This is why it's attracted me.
Okay, thank you. And I ask the audience and another participant if you have any question for the lecturer.
I only have one
if
question.
you-
That who is your supervisor in Debrecen University?
Dr. József Prokés and Ferenc Peles.
Okay, thank you very much. It's a nice work.
Thank you very much.
Okay, thank you very much. Have you any question? If you haven't, thank you for your lecture, and we continue the next lecture titled Examination of Consumer Attitudes Regarding the Consumption of the Novel Foods, especially duck wheats. The author and the lecturer is Dora Szepesi Bencsik. Okay, please start your lecture and presentation.
Good afternoon, everyone. Thank you very much. If it's possible, can you please give me the right? Oh, thank you very much. Okay, so as Anita said, my presentation will be about the consumer attitudes regarding the consumption of novel foods, and especially the duckweed. So, what was the problem? Why I started to deal with this problem and with this group of um, plants? The main problem is the overpopulation of the Earth. As I think everyone knows, the population of the Earth will reach the 8 billion around the middle of November last year. So it causes an increased food demand uh, and, of course, an excessive use of agricultural land sites. So, in short, we must feed a very, very huge amount of people, but we only have one Earth, we only have one planet. So, we want, not just want, we must find sustainable alternatives to solve the food problems, but of course, in in a sustainable way. So researchers started to searching for alternative products to alternative foods for humans, and yeah, sustainability is becoming an increasingly important thing for people, mainly especially for the residents of the developed countries. But sustainability and sustainable development and sustainable food production is absolutely an important thing for all of us uh, on the earth. So, as a possible solution, we can speak about different kind of novel foods. As Professor Banati mentioned, uh, a lot of useful things about the consumption of insect proteins in the plenary session. Uh, we must find new solutions, and duckweed species can be good solutions for solve some problems in connection with food production and in connection with uh, with sustainability goals in the earth. So uh, the aim of my study were to investigate the consumer attitudes towards the novel foods. But firstly, I want to say something about Wolfia species because, as you saw in the title of my presentation, I will speak something about duckweeds. Wolfias are basically in a group of duckweeds. These tiny plants are the smallest flowering plants in the world. Uh, this Wolfia species has long been consumed in Asia as an inexpensive source of protein. They call it kainam, which means water egg. And here in the European Union, two Wolfia species, the Wolfia risa and the Wolfia globosa, are officially considered as novel foods since the December of 2021. So it's a relatively new group of novel foods. And why Wolfia species can be useful for us to solve some problems in food industry and some problems in the environment. Uh, if you see this side of my PPT, you can see that Wolfias contains a huge amount of protein. It's around 20 to 40% is for the dried Wolfias, and around 10 to 20% of stretch, and 1 to 5% of fat, but uh, 60 about 60% of this fat content is polyunsaturated fatty acid, And of course, because these plants are, as you see, has green, a vibrating green color. It contains high amount of lutein and beta carotene, and of course some vitamins and trace elements. But I think the most important that we're able to produce these plants without the using of soil. And I think nowadays, for the sustainable agriculture, it's a key point to don't use more and more soil, and we don't need to use different kind of pesticides the production of Wolffia species. So it can be an alternative to pea, rice, or even the soy protein. So this is why duckweeds and of course Wolffia species can be an important um, species in the near future because of mainly because of these benefits. But as I mentioned, these novel foods are quite new in the European Union, so I think most of the people here don't know so much things about these tiny plants. So I tried to ask the consumers what they know about the Wolffias and what they know about the novel foods in general. So uh, my final goal were to assess the possible product portfolio in connection with the Wolffia species. So during my research, I made a questionnaire, of course, with demographic questions and questions about the novel food consumption and Wolffia consumption. I used Google Forms and I had 190 fillers. About half of them were female and about half of them were male. So let's see my results. Of course, the first question were the age of the consumers. And as you see in this graph, most of my questionnaire fillers uh, were in between 19 and 25 years. But as you see here, more than 21% of the fillers were in between 26 and 35 years. So most of my fillers were young adults mainly. And according to the demographic questions, almost half of them are living in a city or country capital. And my fillers were not from just Hungary, they were from the United Kingdom, from Albania, Serbia, Slovakia, and of course, most of them were from Hungary. I asked the highest education of the fillers. Most of them were from high school. I think this is why mostly our students filled my questionnaire. But more than 28% has now a BSc degree, and more than 18% MSc degree, and some of them have a PhD degree. So my questionnaire fillers were mainly from the young generation um, with high school and university education. And I was curious about the diets of the consumers. Mm, most of them have a mixed diet, eating plant-based and animal-based foods, and some of them signed with this red sign were vegetarians, and they have flexitarian diet around 9%. So most most of them have a mixed diet, and I think how to say, to reduce the ecological the ecological footprint is uh, is a bit harder in case of people with mixed diet. But I think the novel foods can give a good alternative for the consumers have mixed diet or other kind of diets. So I asked the consumers about which following novel foods have you heard about. First one was the noni, um, which is an exotic plant, the chia seed. I think almost everyone heard about it. As you see, uh, more than 184, around, not around, 184 consumers heard about the chia seeds. It's novel food, but you're able to buy it in almost every stores, uh, mainly bio stores and so on. And here are different kind of insects. Um, and as you see, around the half of them heard about the crickets, for example, uh, but in average, the half of the consumers who filled my questionnaire heard about different kind of edible insects. And of course, I asked them if they heard about the Wolfia species, and only 18 people heard about it. I think it's not a big surprise because, as I mentioned, these species are quite new novel foods in the European Union. And my next question were, would you eat novel foods? Because I think, as everyone knows, here in this region and here in Hungary, how to say, consumers have a neophobic attitude. They are sometimes afraid of new things and, of course, new foods. So it was an absolutely relevant question for them. And as you see, most of the consumers answered that yes, but only plants. It was absolutely a good news for me in connection with my Wolfia researches. So with the green, you can see yes, plants and insects too, and I think it's another good news for the sustainable development of food industry, and only just 7.4 percent said that no, I stay away from it completely. So if we see that most of my questionnaire fillers were young adults, we able to say that this age group is not as neophobic as the older ones, for example. So I think in connection with the product development, it's absolutely a good news. And of course, I asked them, "Have you heard that since December of 2021, duckweed species are officially registered as new foods in EU?" And of course, only just 20% said that yes. Mm, and I asked them if you were properly informed about this species of duckweed, would you consume them? Duckweed, to hear the word duckweed here is so the most of the consumers are associating with natural waters and the surface, the green, sometimes disgusting green surface of the natural waters. So I think it will be a hard thing to tell them that most of these species are edible. Uh, so it was a bit surprise for me because most of the consumers say that yes, they can consume these species, and only just 16.8% said that no. So yeah, it was good. It was a good. It was a good news for my research, I think. And as you will see, the majority of the respondents would eat novel foods, but only 58.5% would only eat plants. And of course, most of the consumers not heard about Wolffia species and not heard about that these are novel foods in the European Union. And I asked the consumers in which kind of products they want to try Wolffia species and other kind of duckweeds, of course. Um, Most of the answers were in fresh, in salads, for example, and vegetable creams or smoothies, and of course in capsules as a dietary supplement. So these, I think, according to these results, according to the results of this questionnaire, I think these will be the main fields of product development in connection with wolffias and in connection with duckweeds in general. So I think in the future these tiny plants will became a good alternative for other protein sources and other plant-based food sources, because as I mentioned, we're able to produce them without pesticides and without using the soil. So thank you very much for your attention, and if you have any questions, I try to answer them. Thank you very much again.
Okay, thank you very much. Thank you for your interesting and actual lecture. First of all, I would like to ask a question. Why did you choose the duckweeds among the foods for the consumer's attitude examination?
Uh, because these duckweeds are part of my actual research, and because as I mentioned, these plants are well known in Asia, for example, and these plants can be a good alternative. So I think it's a must to search for new alternatives in food production. So this is why I chose Wolffia species, because they are new, and as I see in the literature and as I see according to my results, um, these are a rich sources of different kind of useful ingredients. So this is why I chose these species.
Okay, thank you very much. I ask all the participants, have you any question for the lecturer? Okay, if you haven't got, we will continue the another authors, the another lecturer. The another lecturer is Anna Molnár. The lecture's title is Heterologous Protein Expression of Mucor Lusitanicus code and HSBA genes in Penicillium, Chrysorgeum, and Pichia pastorus. Uh, I ask, please start your lecture.
Can you hear me and see my presentation?
Yes, everything is okay.
Okay, thank you very much. I would like to welcome everyone. My name is Anna Molnár. I'd like to thank you for the opportunity to speak today. The topic of my presentation is the heterologous protein expression of Mucor lusitanicus, cottage, and HSBA genes in Penicillium chrysogenum and Pichia pastoris. In our experiments, we use Mucor lusitanicus, a model organism in a basic and applied research. This species belongs to the phylum Mucoromycotina and within the order of Mucorales. It's most frequently saprotroph and widely distributed in nature. Mucorales species reproduce asexually by sporangiospores and sexually by zygospores. Mucor lusitanicus is biotechnologically important to produce extracellular hydrolytic enzymes such as lipases, proteases, and organic acids, alcohol, carotenoids, and some of them are used in startle cultures of oriental food fermentations. It is important to note that it can cause post-harvest decay and is also considered an opportunistic human pathogenic fungi causing superficial and invasive infection known as mucormycosis. Mucor rot is caused by the fungus Mucor piriformis, which survives in the soil as spores. We can see decaying fruits became very juicy, and within this juice are abundant spores of the fungus, leading to secondary spread, and the overmature fruit is more susceptible to infect. Mucormycosis is an invasive fungal infection caused by Mucorales species. It can manifest in different forms, and it can also cause gastrointestinal infection by consuming contaminated food. In the fall of the 2013, a large spoilage incident resulted in the national recall of Chobani yogurt, which contaminated with the fungus Mucor lusitanicus. Later studies showed that this isolate was virulent in a murine tailvein infection model, and it was demonstrated the mucor is able to alter the microbiome of the mouse and increase the permeability of the monolayer in epithelial cells. The aim of our research group is to identify virulence factors which is necessary for the pathogenicity of the fungus. Preventing the fungus from entering the healthy tissues, both human and plant, can be a good target. Our research group identified some spore surface proteins as potential virulence factors, such as spore coat protein homologues and hydrophobic surface binding proteins. Cottage proteins are widely present in Mucorales fungi. However, as they are not universal, they have the potential to be targeted for antifungal therapy. Both in Mucor and Rhizopus, cottage proteins are proved to be essential to the virulence. In Rhizopus, cottage 3 protein can bind to the GRP78 receptor and mediates mucormycosis, while in Mucor it is necessary for the spore wall development. The other protein family is the hydrophobic surface binding proteins, as they play a role in the attachment and degradation of components of the plant epidermis and pathogenesis of mucormycosis. Hydrophobic nature of these proteins makes them potential targets because the biofilm formation and adhesion are strongly connected to hydrophobicity. Our experiments aim to establish a highly efficient protein production system in filamentous fungi, Penicillium chrysogenum, and yeast Pichia pastoris. The first step is to construct heterologous expression plasmids that carry gene encoding cottage and HSPA proteins. To achieve the plasmid uptake, PEG-mediated transformation can be used in the case of Penicillium chrysogenum and electroporation in the case of Pichia pastoris. After transformation, the transformants will be selected on minimal media supplemented with selection markers, pyrithiamine for Pichia, for Penicillium, and zeothin for Pichia. To validate the plasmid uptake, molecular biological methods can be used. The next step is the production of the cottage and HSPA proteins by stable mutant strains. It is important to note that we have to clone the expression strain with its own signal sequence into the plasmids to secrete the proteins into the extracellular space. Then the heterologous proteins can be purified by liquid chromatography, and we aim to validate the presence of cottage and HSPA proteins by SDS-PEG and visualized by commercibillion blue and silver staining. Two heterologous protein expression systems were chosen to produce cottage and HSPA proteins. The PREKIA expression system has the main advantages of large-scale production and easy genetic manipulation. The major advantage of the Penicillium chrysogenum expression system is that its filamentous fungi and the closely related species to Mucor lusitanicus. In addition, the protein glycolization and folding is more proper than in Pichia pastoris, and the function of the protein is preserved. It is especially important for us to maintain the activity of the proteins, for example, kinase activity of the cottage proteins. For protein expression in Penicillium chrysogenum, we designed a specific plasmid which containing the cottage and HSPA genes, PEP signal sequence which is required for secretion to the extracellular space, and the PTRA selection marker gene which can provide the selection of the transformants. First, we amplified the 5' UTR with the PEP signal sequence and the 3' UTR by PCR. Then the gene of interest can be fused together with the flanked regions after we perform the double digestion of the expression cassette and the plasmid. Digested fragments were ligated together, and the linearization of the plasmid can be achieved by restriction digestion. This step is necessary for the transformation. We introduced the plasmids to Pichia strain by PEG-mediated transformation. After transformation, individual colonies can be obtained on minimal media supplemented with pyrithiamine. Validation of the plasmid uptake was performed by agarose electrophoresis, where we could detect two cottage expressing strain. After, for liquid fermentation, we inoculated the mutants to gain a large-scale protein yield. Multiple purification steps are necessary to obtain purified proteins. First, for size exclusion chromatography, 50 milliliter bioscale mini BioGel P6 desalting cartridge was used to separate one fraction, which containing the protein of interest. This fraction was further purified by cation exchange chromatography, which allows us to eliminate unspecific components. Then the proteins of similar sizes were separated from our protein sample using S200 HR column. The analysis of purified proteins was done by SDS-PEACH, and the presence of the cottage protein was proven by silver staining and confirmed by mass spectrometry analysis. Meanwhile, we are also working on protein expression in Pichia pastoris. First, we amplified the HSPA1 and HSPA2 genes by PCR. PCR products were ligated into the cloning vector. The insert was cut out by double digestion via these restriction enzymes. The digested HSPA DNA fragments were ligated into the Pichia plasmid. As a result of Pichia transformation, is two HSPA1 and one HSPA2 Pichia transformants were obtained. The HSPA1 positive clones show the expression of desired size protein, whereas the Pichia transformants carrying HSPA2 did not. After size exclusion chromatography, approximately 17.15 kilodalton of proteins were detected in elute 39 and confirmed by mass spectrometry analysis. Our future plans include the optimization of heterologous protein expression in yeast and filamentous fungi for large-scale fermentation of cottage and HSPA proteins, investigation of the activity of the purified proteins, because cottage proteins have presumably kinase activity. To achieve easier purification, histag labeling can be used. To determine the exact cellular localization of these proteins, we already designed GFP-labeled plasmids. For in vitro and in vivo interactions, the purified proteins can be used to determine the role of these proteins during infection. We also aim to investigate the biofilm formation ability in the presence of the purified proteins. And finally, we aim to explore therapeutic application of the cottage and HSBA proteins in the long run. First, I would like to thank the organizers of the opportunity. Support was given by National Research, Development and Innovation Office and Eötvös Loránd Research Network. I would like to thank the head of the Department of Microbiology and all of colleagues who contributed to this research project.
Okay, thank you very much. I asked the participant, have you any question? Okay, if you haven't had any question, we continue with the next lecture. Is the occurrence of deoxynivalenol in winter wheat grains harvested in Hungary over the years 2012 to 2017. The lecturer is Tamás Meszlényi, and the co-authors are Beate Tóth, Dénes Siebert, and Ákos Mesterházy.
Good afternoon, everyone. Can you hear me?
Yes, we
hear me?
can. You
All
can
right.
start the lecture. Please
Okay,
start
we
it.
can hear you.
Right. Today I am going to present a long-term monitoring results for deoxynivalenol occurrence in wheat cultivated in Hungary. In our work, we investigated the trends over time, the regional differences, and the possible effect of climate in regard of toxin contamination. My objective is to summarize our key findings of our analytical work. As you are all aware, wheat is the most important small grain cereal crop in the world. As a staple food, it is the main source of calorie and protein for an estimated 35% of the world's population. However, it is also appealing to several pathogenic microorganisms. There are over 200 different disease-causing agents which are threats to wheat production. Fusarium head blight is considered as one of the most destructive one, which can caused by about 15 to 20 Fusarium species. The local species distribution depends on climatic factors, but it can be stated that in general, the wet and moist conditions during flowering promote the development of this disease. And globally, Fusarium graminearum sensu stricto is recognized as the predominant one, but Fusarium culmorum and Fusarium avenaceum are also commonly recorded from infected plant tissues. This disease can cause yield loss, quality reduction, and mycotoxin contamination. And although epidemics occur about every fourth year, their economical damage can be huge. In some years, it can be billions of dollars in losses. Among the mycotoxins associated with this disease, DON is the most important one, which constitute a serious hazard to human and livestock when it is present in above limits in foods and feedstuffs. Contaminated foods can cause, for example, growth retardation, gastrointestinal dysfunction, and autoimmune diseases. To preserve the health of consumers and livestock, maximum limits were established. These regulatory levels depend based on toxicological and exposure data. In the states of the EU, the maximum limits for DON in unprocessed cereal-based foods is 1.25 milligram per kilogram, and in baby foods it is just 0.2 milligram per kilogram. The problem is that because of the climate change, the frequencies and intensities of the epidemics will change, and with the more frequent extreme weather events, the fungal distribution and again the epidemics will also be affected. So for this reason, continuous monitoring on the mycotoxin occurrences and constant review on existing regulatory levels is also necessary. But however, there is a lack of national monitoring programs out there. So the results of measurements established by accredited laboratories have a real value. And we have some such data. So the evaluated toxin levels were measured by accredited laboratories of SGS Hungary Ltd using HPLC method. Between 2012 and 2017, they analyzed almost 6,000 unprocessed winter wheat grains. This table shows the number of samples tested in each year. I have to note that these samples were mixed and stored, but we don't have information regarding the length of the storage period. So it is possible that some of the samples were measured 1 year after the harvesting. And I also have to mention that the toxin data that we got were grouped by county. So we have average contamination levels. To investigate the relationship between toxin contamination levels and the climatic conditions, we downloaded the weather data from a Hungarian website called MetNet. And we use the data of May for these reasons, as the wheat is most susceptible for the fungal infection at flowering. So in my first table, you can see the mean DON concentrations per year per county. The values that surpass the previously mentioned 1.25 milligram per kilogram are highlighted with red. As you can see, there are just 3 of them, so we can state that the toxin contamination levels were generally low in the studied period. But if we take into account and take a closer look, we can see how many concentrations are there which exceeded the limit for baby foods. So we have to say that these values aren't insignificant. In the last rows, the mean concentration values per year are shown. In the year of 2015, the concentration level were significantly higher than in other years. But no other significant differences were found between other years, which is in accordance with the observation that epidemics occur just in every 4th year. These maps shows that in the year of 2015, the precipitation levels were higher than 2012, and also the concentration levels were higher. So we think that the reason behind the higher toxin contamination levels is the much more rainfall. In this slide, you can see the maximum levels per year per country. You can see that there were 2 years where the means, but the maximum levels exceeded the 1.25 milligram per kilogram. The highest level measured was the 5.42 milligram per kilogram, which is almost 4 times higher than the limit. Here again can explain the higher toxin levels by the rainfall levels. The precipitation levels were higher in 2016 than in 2017. So we have looked at the differences between years, but what can be said about the regional differences? Here you can see the mean data grouped by regions. Based on our variance analysis tests, there were no significant differences between the counties. But unfortunately, as you can see here, there were some missing values in cases of the earlier years. So we only involved the results of the last 4 years in our further tests. For the illustration, here you can see the grouped data with the Hungarian map behind them. At first sight, it seems that the southern part of the Hungary showed higher toxin contamination levels. The highest levels was detected. The samples collected from here. But according to our results of the two-way variance analysis, in regard of the geographic distribution differences, only significant differences was found between the southeast and northeast, and between the northeast, northwest and southeast region. We should also mention that the toxin contamination levels were generally low, but higher occurrences anywhere can happen. And the generally low contamination levels were maybe due to the improved practices, plant protection practices of larger producers. And in conclusion, let me sum up my main points. Continuous monitoring of mycotoxin contaminations is necessary because their results contribute to the development of predictive models and potential adjustments of existing regulatory levels. It is prompted by the climate change as unpredictable extreme weathers and unpredictable epidemics go hand in hand. Establishing predictive models requires detailed info, such as inside meteorological data, and we also should have known the origin, the variety of the cultivars. So as I mentioned, in the studied period, the contamination levels were generally low. And I have to note that the cheapest way to prevent larger economical damages is to use cultivars with higher resistance levels and breeding for resistance. And one last thing I would like to mention is that there is an available article written by my coworkers which evaluated the toxin data of the maize samples. And also I would like to thank SGS Hungary for making their results available to us. And thank you for your attention.
Okay, thank you very much for your lecture. I think it is an important topic in agricultural practice, and I would like to ask why you can examine the deoxynivalenol and will you measure another toxin concentration of the wheat in the future?
So these values actually originated from SGS Hungary Ltd, and the grain samples originated from farmers all over the regions of Hungary. So the measurement wasn't actually carried out by us, but the company. They just made available to us the data to evaluate the data. Their measurements are continuously go.
Okay, thank you very much. Have you any question?
I have also one question for you. Only you have data about this presentation you show us that on a DON toxin,
Yeah.
the limit. And did you check about that the farmers that you get the samples that what kind of fungicides they were using or not?
Sadly, I don't have information regarding to that.
Okay, no problem. I just wanted to ask because maybe it would be later on a good idea when they are doing that. You can check what kind of fungicides they were using and which one. Maybe that one that had a high limit of the DON toxin. Maybe they didn't use specific fungicides. So maybe later on it will be a good.
Absolutely. You
Yeah,
have a point right there.
yeah. Thank you very much. It
Thank
was
you.
very good.
Okay, thank you very much. And we continue with another lecturer and author and another topic. The next lecturer and author is Csaba Vasko, and his topic is sustainable rural development through the utilization of invasive alien crops to produce local bioenergy. Please start your presentation.
Thank you. Thank you very much. First of all, I would like to thank the organizers very much for giving me the opportunity to speak here or present here today. In my professional life, I'm a consultant. I work as a consultant specifically on climate change, energy, and land management and water resource management issues. And I'm not going to talk about food. I'm going to talk about bioenergy. And what I would like to share today is a selected list of experiences of working together with rural communities on local bioenergy production and better ecosystem management. So this is really a little bit learning by doing. I hope my presentation moved to the next slide. Yes. So my primary objective was to set up a market-based mechanism where that can provide benefit, socioeconomic benefit for local communities, but also can contribute to biodiversity conservation. So the project or the experiment was based on the idea of using invasive alien plants, more precisely shrubs, invasive alien shrubs, as bioenergy source to produce local bioenergy to replace natural gas and to reduce energy poverty. But the project also has other impacts. In addition to energy production, the project generated new new employment skills, and or improved employment skills and biodiversity, and it has also improved a little bit, at least at pilot scale, the flood security. Yes. So my research area is northeast Hungary. The floodplain of the River Tisza, which is the second biggest river in Hungary and the very important ecological corridor between the Carpathians and the Danube. And the key problems that I addressed or that I detected is basically that the key land and water use driver is agriculture, which is mainly intensive here. And it was the driver of the mismanagement of the floodplain areas. The river was regulated, channeled, and the floodplain was narrowed between the dikes. And most of the ecosystems were converted to croplands. And the vulnerability to climate change is also very high. So farmers and communities are exposed to unpredictable floods, and today's recently rather more unpredictable droughts. The stresses arising from climate change are amplifying the impacts of human stresses. So there is energy poverty, for example, and migration from this region to other cities, to bigger cities. So all this has led to a broken balance between people and the nature. So the deterioration of floodplain ecosystems. And the symptom that I addressed and what I'm working on is basically the expansion of invasive alien species. I don't want to go into the details of basics of invasive alien species. I just would like to emphasize that invasive alien species cause serious economic damages, usually agricultural and forest management or related to forest management, but they also cause human health problems, primary reproduction problems, habitat degradation, even extinction, and they can even modify local hydrology conditions. The socioeconomic cost caused by invasive alien species is estimated to be at least 12 billion euros in Europe. In our case, in this case, Amorpha fruticosa is the particular invasive alien plant. More precisely, it is a shrub that was introduced from North America as an ornamental plant. It was used to also to stabilize sandy dunes, and but it has been expanding, especially on the floodplains, very aggressive. The land use 50 years ago on a certain section of the River Tisza floodplain, the land use was mainly grassland management, meadows and grasslands. And on the right side of the slide, you can see the current land use system, which is more dominated by cropland management. And the red circles represent the location and the size of Amorpha fruticosa infection. And on this section, 23% of this section is basically the land use, or the land coverage is basically Amorpha fruticosa, or infected by Amorpha fruticosa. Yes, this is just a summary slide of the harmful impacts or effects of the rapid invasion of invasive Amorpha fruticosa. Their expansion is leading to habitat loss and habitat fragmentation. So the biodiversity value is very, very low. They literally colonized certain sections of the floodplain, which means that the density is very high. It is relatively tall, and they function as a bottleneck when the flood is coming. So the water level is higher upstream than downstreams. Regarding land management, this is basically, I mean, the eradication is an extra task for farmers and forest managers, so they have to spend extra money to eradicate basically every year this invasive shrub. On the other hand, it is a very promising energy source, which means that it was proved to be a good energy source if we only look at the moisture content of different biomass resources, which is not the most important factor, but a very fundamental component of biomass potential. We can see that the 24% moisture content is, as compared to other biomass resources, is very good, especially that taking into consideration that this is the moisture content at the time of harvesting. This shouldn't look like that. This is basically there is a background photo, which is a floodplain section, the orthophoto of the floodplain section. But this slide represents or shows the process of the land use, new land management, the introduction of new land management. In brief, a local community started to eradicate the Amorpha fruticosa, and they transported it and processed it into wood chips, and the wood chips became a product, and the product was sold either to a power plant or a heating plant, or it was used locally in local public buildings. We did not stop at this point. It was highly important for us to introduce new land management or new land use after the removal of invasive shrubs. Depending on the local conditions, this new land management was either grassland management, semi-natural grassland management by domestic animals, or in former coal plants, it was native energy useful tree plantations, which provides or will provide the future biomass after the utilization of invasive shrubs. Just one slide on the native willow plantations. That's one of the new land management that we introduced in this local community, in this floodplain. These are composed of native willows, so nothing really special. And unlike traditional or intensive agricultural practices, we implemented heavy tillage only in the first year, in the year of planting. So there is less impact on the soil, and the plantation, depending on the age, it provides biomass every 2 or 3 years. However, the moisture content is much higher as compared to Amorpha fruticosa, but that is the future biomass. Again, there should be this is not an animation, but there should be an orthophoto of a village. Of course, it was very important to identify and calculate the local energy demand. So try to imagine there is a orthophoto of a small village, the center of the village. Originally, we planned to establish a central village heating system, but due to the high investment costs, we ended up in establishing or installing individual boilers in local public buildings. And since the public institutions are located close to each other in the village center, at least the logistic cost became a little bit lower. This is basically the model of the initiative or the interventions. So it was set up in a way that the invasive shrub is eradicated, harvested, collected, and converted into wood chips. And the wood chips is was either sold to a power plant to test it and use it, or a district heating plant, and later it was used by the local community to heat local public buildings. I would like to emphasize that the biomass in the first 2 or 3 years is Amorpha fruticosa, but later on the main biomass resource was and is becoming the biomass from willow plantations. This slide shows and represents the impacts of the project, at least at pilot scale. I think good experience is linking and the energy poverty reduction with conservation, nature conservation, and flood security benefits. And finally, a few selected key conclusions. So the expansion of invasive alien shrubs has increased and is increasing flood risk, and it's reducing biodiversity, and it's increasing land use costs, land use efforts. Amorpha fruticosa was proved to be a good biomass source. It has been tested, and it is a good biomass source to produce heat energy in public buildings, to replace natural gas, and eventually to reduce energy poverty level in local public institutions, public buildings. But Amorpha fruticosa as an alien species can be or should be used only in the first 2 or 3 years, so only the first harvest, and it should be followed by, and it is followed by native biomass from native willow plantations. And finally, which is very important from our perspectives, is new land use interventions are needed to prevent the regrowth, the reshooting of invasive shrubs, and this can be grassland management, so grazing, energy plantations, and water cover. Thank you very much. Thank you very much for your attention, and I'm happy to take questions if you have any. Thank you.
Okay, thank you very much. It is one of the most actual topic today's, I think. Have you any question? No, but I have a question. What do you think? Have you any other alternative source of bioenergy? And which is the most important research topic and important research question that you suggest, or you will continue for this research topic? What do you think? What do you suggest?
So you're asking what is alternative biomass resources? That was the first question.
Yes, or another important questions, research topic to continue this topic.
Land ownership conditions, because that is a critical factor when we start these kind of project, that the land ownership conditions has to be, and land ownership conditions have to be very clear. It is really matter that who owns the land that we would like to, if we aim to intervene. And the clarification of this question might take years beforehand, but the floodplain is a very good territory to start to implement and using invasive alien species. I'm focusing on that. And the other critical question is basically what is coming afterwards. So what kind of new land management new land uses and new land management methods can be utilized after the removal of invasive plants, and there has to be a cost-benefit calculation because it has to be maintained continuously after the eradication of Amorphophuticosa. There are many attempts, especially along the River Tisa and the River Körös, that many enterprises are trying to utilize invasive alien shrubs from the floodplains, but there is no new land use management after the eradication, and that is critical from flood risk, flood security point of view, nature conservation point of view too. Thank you.
Okay, thank you very much. And we would like to continue with the short 5 minutes long presentation. The first short presentation titled is Evolution of Water calthrop harvested from Lake Tisa in Hungary. And the lecturer is Judit Tarak Tiliszczak, and the co-author is Gyula Kücsön. Please take your presentation.
Thank you. Dear participant, let me introduce my short presentation entitled Evaluation of Water calthrop Trapanatans harvested from Lake Tisa, Hungary. Water calthrop is an annual aquatic weed. The plant has floating rosette, which produce a large woody fruit with 4 barbed spines. The fruit develops underwater. When mature, the fruits fall from the plant and sink to the mud. A single seed may give rise to 10 to 15 rosettes, each producing up to 15 to 20 seeds. Water calthrop is very competitive with desirable aquatic species in shallow waters with soft, muddy bottoms. It can develop nearly impenetrable mats across wide areas of water, which can block light penetration, reduce oxygen levels, leading to fish kills and impact fishing and recreational activities. The fruits drift to shore, where their sharp spines may hurt bare feet. Our aim was to study the water calthrop fruit as food material. Water calthrop plants with fruits were harvested manually from Lake Tisa under official permission, since water calthrop is a protective species in Hungary. We made a chemical and a physical characterization of the fruit and minimal processing on the fruits. With the applied minimal processing techniques, we separated the liquid and its solid content. Yield, dry matter, soluble solid content, and color were measured on the separated biomass. The kernel's macrocomposition and inorganic elements content, including heavy metals, were analyzed. The water calthrop kernel is a good source of bioactive substances. The wet kernel didn't contain heavy metals, arsenic, mercury, palladium, and cadmium. It is a source of potassium. 500 grams of wet kernel can provide the daily potassium intake for the human body, and half of the zinc, a quarter of the copper and iron intake for the human body. The water calthrop whole kernel flour, this is a dried formule, has a macrocomposition of a whole cereal flour. It has a 12% protein, 2.5% ash, and 70% total carbohydrate content and rich in fiber. The whole fruits were selected by size and weight. The manual sliceability and woodiness of the fruits were determined after 24-hour 1% cellulase enzyme treatment. The fruits need to sort by size for economic processing. Smaller and lightweight fruits, up to 1 or 2 grams, can be measured easily with knife mill. The fruits in the weight range from 3 to 5 grams can be applied for manual cutting. If the fruit weight is above 6 grams, it is necessary to use cutting machine. The dehulled, fresh, rinsed kernel was screw pressed, then the juice was clarified by centrifugation. The half of the water calthrop kernel is moisture. It doesn't increase if the rinsing water is properly removed. The pressed kernel juice has a 25% dry matter content, while the pressing residue has a 37% dry matter content. With centrifugation, the ratio of the wet sediment was 40%, and the soluble solid content of the supernatant was 6%. The juice and the pomace color didn't change within a shorter time during lab processing and measuring at room temperature. We concluded that the water calthrop fruit harvested from Lake Tisa is suitable for food utilization. The fruit kernel has low fat, high carbohydrate, fiber, and potassium content. Safe food ingredient and suitable for developing food specialties, for example, vegetable milk or fermented drinks, or for dough fortification as fiber aid ingredient. Thank you for your attention.
Thank you very much for your lecture. It was one of the most interesting lecture about a very special topic. I asked the participant, have you any question for the lecturer? Okay, if you haven't got any question, we will continue the next topic, the investigation of tennis activities in Mucormycosa fungi. The lecturer is Tamás Kovács, and the co-authors are Anita Kovács, Csaba Vágvölgyi, Judith Krisz, and Miklós Talko. Please take your presentation.
I think just we have to wait a little bit because I can see there is some connection problem with Tamás.
We are waiting some minutes. No, we can't. Don't hear you.
Yeah, Tamás is writing that sorry. I don't know what the problem.
Yes, I see. I suggest we continue with the next lecture, and Tamás Kovács can take your presentation at the last. Okay.
Okay, akkor next one is Norbert Magyar.
Right. Okay, the next short presentation titled Application of Data Analysis Methods in Determining the Migration Kinetic of Polymer Additives. And the lecturer is Norbert Magyar. Please start your presentation.
Okay, dear chair, dear participants, please let me introduce myself. As the chairwoman said, my name is Norbert Magyar. I work for the Budapest Business School, Faculty of Commerce, Hospitality and Tourism. I would like to give you a short presentation on our study about migration additives of polypropylene-based plastic into food simulants. The research was carried out in a cooperation with the Joint Research and Training Laboratory on separation techniques. It works at the Eötvös Loránd University. I was one of the statisticians in this group. In this study, the migration kinetics of food plastic additives from polypropylene, which is a food contact plastic, was investigated. The correlation between swelling kinetics of polypropylene in isooctane and ethanol food simulant and migration kinetics of 6 food contact plastic additives into these simulants was analyzed. We determined the equilibrium time based on migration kinetic curves of these additives in order to optimize the contact time of food contact plastic with these food simulants. We also observed the maximum migrated concentration of the plastic additives in isooctane and ethanol to determine which simulant, so which one out of these two is more appropriate for polypropylene. The plastic sheets were cut into small test specimens. The geometry of them, so the width, the length, the height, and weight of each specimens were measured before immersion, and the samples were stored at the lab on 40 degrees in an incubator. The contact times for the kinetic studies were 5 minutes, 30 minutes, 60 minutes, so 1 hour, 2 hours, 6 hours, 12 hours, 24 hours, and after 1 day, we had daily samples from 1 day to 13 days. Okay. So based on the swelled and dry weight, we can determine the swelling degree from this mass amount. So the swelled minus dry over dry give you the swelling degree. Based on the measured dimensions, the surface area of each specimen was calculated, and the result of quantitative analysis with gas chromatography gave information to us about the mass concentration of the additive food simulant, of course. And what it's really new in this study is the application of variography. So empirical semi-variograms were calculated and we plotted them to assess the temporal autocorrelation structure of the concentration of migrants and swelling degrees. So as I mentioned, it's a geostatistical tool, which sounds a bit strange, but we applied first by our team on the field of chemistry. So with the use of variography, the necessary contact time was determined to reach the equilibrium. The relationship between the swelling of the plastic and the additive migration was investigated using a Pearson linear correlation test. And besides this, the maximal migration concentration of plastic additives in isooctane and ethanol, so the given these two food simulants was also determined. The significance of the differences was tested using analysis of variance, and the normal distribution of the data was verified using the Shapiro-Wilks test. It's a general test to test the normality, and the homoscedasticity assumption we assessed was assessed by Barset test. So due to the absence of homoscedasticity, Wilks ANOVA was used, after which Games-Hewell test, post-hoc test was applied to compare all possible pairs of additives. The same analysis was performed, of course, on the swelling data, so not just on the concentrations. Okay, in the case of BHT, that's just one additive, but I have 5 minutes, so I would like to show just this. 120 hours were determined as a necessary contact time. This is a variogram, a semi-variogram, on which you can see that around 120 hours, we have the level on which the points varies around the variance. Okay. Just a second, I try to open the next slide. And as the molecule weight increasing, we obtain that. So as the molecular weight of the additive increases, the concentration decreases. Of course, the migrated concentration decreased. As a conclusion, it can be stated that the swelling degree of polypropylene is significantly higher in isooctane than in ethanol. There is an excellent correlation between swelling degree of plastic and the migrated concentration of examined plastic additives. The migrated concentration of the plastic additive depends on its molecular weight, and the time for reaching equilibrium depends on function of plastic additive, the molecular weight, and the type of the food simulant. And it's not necessary to carry out migration tests in ethanol. You could see that in isooctane is higher. The concentration is higher. So thank you for your kind attention.
Okay, thank you very much for your presentation. I ask the participant, have you any question?
Sorry, I don't have any question. I just want to ask whether you can hear me now this time.
Yes, okay, Tamás.
Yes, thank you so much.
Yeah, we can hear you. Okay, maybe we can continue with Tamás, Anita, yes?
Okay, we can continue with
I just
Tamás,
wanted to ask
if you would like.
because...
Okay, thank you very much. We will continue the next lecture. The lecturer is Tamás Kovács, and the title of the lecture is Investigation of Tannins Activities in Mucromycota fungi. The lecturer is Tamás Kovács, and the co-authors Anita Kovács, Csaba Vágvölgyi, Judit Kiss, and Miklós Tákó. I ask, please take your presentation.
Thank you so much. Welcome everyone. I'm Tamás Kovács, and the title of my presentation is Investigation of Tannins Activities in Mucoromycota fungi. Tannins is an inducible enzyme that are produced by bacteria, yeast, and filamentous fungi. It can catalyze the hydrolysis of ester and dipside bonds that are present in complex tannins, gallic acid esters, and hydrolyzable tannins. It is mainly utilized in the food and pharmaceutical industries. In the food industries, it is mainly used as a clarifying agent, an agent that can reduce bitterness of different beverages. And in the pharmaceutical and chemical industry, it is mainly used to produce gallic acid, which is the intermediary compound in the synthesis of trimethoprim and propyl gallate. Many members of the Mucoromycota phylum are known as an excellent producers of extracellular enzymes. However, their tannins production is barely studied. Therefore, the aims of our work was to isolate tannins-producing strains from the Mucoromycota fungi to produce enzyme in solid-state fermentation and submerged fermentation, and to measure the tannins activity with colorimetric spectrophotometric method. We selected 7 potential strains from the Szeged Microbiological Collection for analysis. in which their tanners production was tested on multi-extract agar plates that was supplemented with tannic acid as an inducer. Only 5 strains were able to tolerate the inducer and had tanners activity in detectable level. The tanners production of these 5 strains were further examined in a wheat bran-based solid-state fermentation system and in a submerged fermentation system where we used yeast peptone broth. During both fermentation, tannic acid was used as an inducer, and there were also control systems without tannic acid. The tanners activity was measured using the methanolic rhodanine method, where gallic acid is released from their substrate, and rhodanine can specifically bound to gallic acid to form a colorful complex, and this color intensity is proportional to the tanners enzyme activity. We expressed the results as unit, and one enzyme unit was defined as the amount of tanners enzyme which produces 1 micromole of gallic acid per minute. As I mentioned earlier, only 5 strains were able to produce tanners on multi-extract agar. You can see those 5 strains. The clear zone on the agar plates is due to the hydrolysis of tannic acid due to tanners activity. During solid-state fermentation, all 5 strains were able to produce tanners, but only in systems that contain tannic acid. So we can conclude that the presence of an inducer like tannic acid is necessary for tanners production. The highest enzyme activity was measured in case of Rhizopus microsporus varietas oligosporus, and we also can see that in the systems where there were no tannic acid, there were no tanners activity in detectable level. This also can be seen in case of the submerged fermentation, since no tanners activity can be seen in the system without tannic acid. We also can see that the higher tannic acid concentration results in higher enzyme activity, and the highest enzyme yield was measured in case of Rhizomucor mihei after 7 days of incubation in the systems supplemented with 0.5% of tannic acid. According to our results, some members of the Mucoromycota phylum are capable of producing tanners, but only in the presence of inducer. Tannic acid was proved to be an excellent inducer of tanners production. Rhizopus microsporus varietas oligosporus resulted in the highest tanners activity in the wheat bran solid-state fermentation system, and Rhizomucor mihei was the best tanners source in the submerged fermentation. And comparing the results of both fermentation, we can conclude that higher enzyme activity can be achieved in the submerged fermentation systems. And thank you for your kind attention.
Okay, thank you for your presentation. I ask all the participants, have you any question for the lecturer? Okay, if you haven't got any question, I suggest we continue with the next lecturer, Lauren Senado, with the biocontrol of aflatoxin contamination and the effect of Aspergillus flavus on the maize quality. The co-author is Tunde Pusztahely. I ask, please take your presentation. Sorry, I can't hear you. We can't hear you.
Hello.
Okay.
Hello. We can hear you. You can start it,
Sorry
okay?
about that. Yes, hello everyone. Good day and thank you for the opportunity. My name is Lauren Senado. I'm a final year master's student from the University of Debrecen, and I'll be doing a presentation on the biocontrol of aflatoxin contamination and the effects of Aspergillus flavus on maize quality. My work was supervised by Professor Dr. Tunde Pustahili, also from the University of Debrecen. So maize is ranked as the world's second most important cereal crop due to its nutritive, its valuable, and its phytochemical compounds. And however, maize is contaminated with secondary fungal metabolites known as aflatoxins, which is mainly produced by Aspergillus flavus. Toxinogenic fungi, and then biocontrol. And we also have environmental conditions such as precipitation, moisture, pH, climate change, among others, affects the level of aflatoxin B1 contamination. But however, this contamination does not only affect the health of human, that is, it does not lead to only chronic and acute effects, but it also affects the grain quality, that is the protein and the starch content. However, this effect may vary due to several factors. It also leads to huge economic losses in most countries. Therefore, aflatoxins have become a major concern in this world. Now, the main objective of this study was to assess the effects of toxinogenic and atoxinogenic fungal contamination on the nutrient value and mycotoxin content in maize. And in the materials and methods, the main study site was the University of Debrecen, and it was carried out between the years 2020 and 2022. And the treatments that were used for the Aspergillus flavus was toxinogenic and atoxinogenic, respectively. And in the data collection, aflatoxin B1 contamination was carried out using the HPLC technique. And also we have the grain starch and then the protein content, which was carried out using the classical method. And the main statistical analysis that was used in the study was the GenStat. Now, the results indicated that Aspergillus flavus contamination did not significantly affect the kernel, that's the protein and the grain starch content. Now, this is because looking at the first graph that's on the starch content, contaminated Aspergillus flavus was 61% as compared to 63% in the control. And then when we look at the protein content, it was contaminated Aspergillus was, and then control was close to 12%. So it did not significantly have any effects due to the several factors that we mentioned. That's looking at the environmental conditions such as the climate change, looking at the precipitation, water, etc., didn't have any adverse effects on it. So in conclusion, we'll say that the study concludes that Aspergillus flavus incident did not show any effects on maize grain quality. Thank you for your attention, and any questions are welcome.
Okay, thank you. Have you any question for the lecturer for this topic? Okay, thank you very much. We haven't got any question.
Thank you.
Thank you. And I suggest we will continue the next short lecture. The title of the next short lecture, The Impact of Lactic acid and ascorbic acid mixture on the quality parameters of wild boar meat. And the lecturer is Munknasan Engbold, and another co-authors are Attila Lőrinc, majd Elayan László Friedrich, Attila Solymosi, and Balázs Vízt, Jani Kornél, and Adrián Tóth. I ask, please take your presentation.
Thank you. Good afternoon, everyone. Can you hear me well?
Yes, we can hear you very well.
Okay. I hope you all enjoying and learning a lot of things from today's conference. Briefly about myself, my name is Mukna Sanyihpold. I'm from Mongolia. Currently, I'm a PhD student at Hungarian University of Agriculture and Life Sciences. So today, my presentation topic is the impact of lactic acid and ascorbic acid mixture on quality parameters of wild boar meat. Here you can see the table of contents of my presentation, introduction, material and methods, which were used for our research, results and discussion, and the last one is conclusion part. So let's start. The objective of the current research was to evaluate the effect of organic acid in European wild boar meat and investigate the aging procedure in vacuum packaging. Wild boar meat is reported to contain a lower concentration of fat and cholesterol than meat from the domestic pig. Wild boar is species that is hunted for food and sport all over the world. The main problem causing shelf life of quality of the meat and meat products are microbial growth and oxidative rancidity. Biopreservatives can help increase the shelf life of food by lowering pathogenic bacteria survival and improving overall food quality in inhibiting oxidative rancidity. So let's jump to experimental steps. For sample, we used wild boar meat from the local processing plant Wadex. The meat cut were packed in low density polyethylene pouches and were transported to the laboratory under chilled condition and stored in 4 plus minus 1 degree in 1 day. Then we started our experiment. The samples were cut into stacks of similar sizes. Half of the samples were immediately vacuum packed for the control sample, and the rest of them were treated with organic acid mixture. The treatment was performed by spraying method. The mixture of 2% of lactic acid and 2% of ascorbic acid was applied at the room temperature on both top and bottom surface of the meat samples. Samples were vacuum packed after treatment, and after vacuum packaging, samples were stored at 4 plus minus 1 degrees. The packs were stored for up to 21 days, and quality parameters were measured on day 1, day 7, day 14, and day 21 in every week. And we measured pH, water holding capacity, water activity, and microbial evaluation, aerobic total germ count. Now, most important part, results. And the value of the pH of wild boar meat treated and non-treated samples are shown in the table. During the 21 days of storage, the pH of samples treated with 2% of lactic acid and 2% of ascorbic acid mixture and non-treated control samples all significantly lower than day 1 of the storage, and lactic acid and ascorbic acid mixture decreased the meat pH by about 0.07 unit through in day 1. It is due to organic acid treatment. The measured pH of the solution was 2.48. That's why it's lowered. But at the end of the experiment period, the pH of treated sample was slightly higher than non-treated ones. About water activity and water holding capacity. During the storage period, water activity and water holding capacity of treated and non-treated samples were increased. And on the day 21, treated samples were slightly higher than non-treated samples. pH has high influence on water holding capacity, which is closely related to product yield and meat quality. Water holding capacity is ability of meat to retain its water during processing, storage, and cooking. So low water holding capacity often results in high drip loss and poor eating quality, like drier, tougher in cooking stage. Microbiological evaluation result for this study indicate that the treatment of 2% lactic acid and 2% ascorbic acid mixture had significant advantage to maintaining shelf life. Initial aerobic plant count of non-treated samples were significantly higher than treated samples in the day 21. So in a conclusion, lactic acid and ascorbic acid treatment may be an alternative to extend the shelf life of wild boar meat. So at the end, I would like to thank to my supervisors and colleagues who participated in this research, and thank you so much for your attention.
Okay, thank you very much. And I ask, have you any question for this topic? No? If you haven't got any question, we continue with the next lecturer, Priti Lanta. The co-authors are Péter Tamás Nagy and Tamás Magyar, and the title of the presentation is a Statistical Evolution of Physical and Chemical Parameters on microalgae cultivation in different nutrient solutions. I ask, please take your presentation.
Okay, hi, welcome everyone. So the topic is statistical evaluation of physical and chemical parameters on microalgae cultivation in different nutrient solutions. So yeah, so we all know what circular economy is. So initially we had linear economy, that is that we used to take the things, we used to make whatever we wanted, and then we used to waste. But in circular economy, we take, we make, and then we try to recycle whatever we have used. So our the research that I have done focuses on circular economy. So the microalgae that I use is Chlorella vulgaris. We all know the applications of Chlorella vulgaris. It is used as a protein supplement, and you can find it in bio shops. It's also used as an animal feed. It is used to treat wastewater treatment plants. So if you add microalgae, it can treat the wastewater treatment plants. It is also used to make a biofuel. So let us see the statistical evaluation. So first, in order to grow the microalgae, we used these different things. The first thing is Atox BBE. This is an atoximeter. In this, as you can see, this green thing, microalgae was put, Chlorella vulgaris, and this atoximeter, it measures. Here you can see the screen. It measures the photosynthetic level. It also measures the toxicity level. It measures the temperature. So all the biological temperature parameters were measured through atoximeter. And then nanocolor and visocolor, these were the two things used for finding out the different chemical parameters like ammonium, phosphate, nitrate. We also use double beam spectrophotometer in order to find out the absorbance, and this is the photometer. So first, let us see the nutrient solutions that were this is the nutrient solution A, AL, T3 and T3L. So A, AL were used, the nutrient solutions that were used in A and AL were the were the solution that was given through us by the company, what we used, BG11, and T3 and T3L were the modified nutrient solution that we made by adding some extra amount of phosphate and nitrate. L denotes aeration. So this nutrient solution was aerated, as well as T3L was aerated, while the others were just added. So this is a chemical composition for A, that is Atox nutrition. The chemical by the company BG11. So this was added in the nutrient as a nutrient solution. So now let's parameters that we found. So when the microalgae started growing in A sample, you can see that the chemical parameters have started decreasing week by week. So you can see a high decrease in nitrate. So initially the nitrate was huge, but as you go down and as the weeks pass by, the nitrate is decreased. Well, as the other chemical parameters, you can see a steep decrease in them. The brown one is phosphate, while the yellow one is COD, and this is the ammonium. So you can see that as the weeks passed by, the chemical parameters were decreasing. This showed that the microalgae was using these chemical parameters in order to grow. And this is the graph that we found through Atox. So you can see that initially till week 3, there was a huge growth in the algae. And then suddenly, due to inadequate temperature, as the temperature started dropping, you can see that the growth of microalgae also has stopped to drop. It has dropped a lot. So here you can see this is the graph that we found through the spectrophotometer. So as it is found that the blue line, that is this, is the week 3. So you can see, as I showed you in the last graph, that week 3 has the highest growth level. And here you can see that until week 5th, the growth of microalgae has, the Chlorella vulgaris is already dead. It's not growing anymore. So as we saw in the graph, till the 5th week, the Chlorella vulgaris was already dead. This is the Pearson correlation table. So there's nothing more. It's just that the chemical parameters, they show a positive correlation with the growth of microalgae, and whereas pH and EC, that means lower the pH and lower the electrical conductivity, that was not good for the growth of the microalgae. Now let's go on to AL. Now AL is the same nutrient solution what the company gave us, that is BG11. It was just aerated. So there is no major difference in the graph about the chemical parameter to the biological one. So you can see that the biomass is way more higher than just A. So you can conclude that aeration is actually good for the growth of microalgae, because you can see a higher amount of biomass in this. And also that also after some time, because of inadequate temperature, there was less growth. But suddenly due to the aeration, the growth of microalgae also was increased. That is what we observed in our evaluation. Yeah, these are the same graphs, and you can see the picture. This is AL, that is aerated BG11. Until the 8th week, the color of this is yellow, so it grew up till the 8th week. Yeah, here is the Pearson's correlation. It's kind of the same. It has just the numbers differ. It has a positive correlation with all the chemical and biological parameters. That means the more the chemical things, the more growth, and also a negative correlation with pH and EC. Yeah, so this is the T3, which is the modified BG nutrient solution that we used. This is the stock solution for that with all the chemical parameters. I won't go in detail to say about everyone. You can just see it on the slides. So this is a modified BG11 nutrient solution that we made with increased amount of nitrate and phosphate. So let's see the graphs of this too. So you can see that there is an increase in this is T3, so it was without aerated. So initially there was growth, and then suddenly because of increased phosphate and nitrate, you can see that the biomass level has increased up to a great extent, even during the week 5th, 6th, and 7th. So there was a highest increase during the week 5th. So this is the picture during the 7th week. So you can see that it's still kind of green, and it was still living because of the increased phosphate and because of the increased phosphate that we added and nitrate. So this is the same Pearson's correlationship. I want let's see T3L. So T3L we found out was the best solution because it created and it was modified by us. So you can see it is a constant and stable increase. There is no like major deviation, no steep increase, no steep decrease. And there has always been a stable increase in T3L because of the aeration that was done. So you can see that this is the during the week 4th, there was a highest growth. And yeah, this is a picture of it during week 7th. And you can see that it is still, it wasn't yet dead, whereas AL were already dead. And here you can see the absorbance. So here you can see that during week 6, there was the most growth. And this is a Pearson's correlation chart for it. So now let us just see the visual evaluation. So this was week 1st when we added microalgae in each of the nutritions. So as you see, as the week passes by, during week 4th, you can already see that A and AL has started to turn yellow, while T3 and T3L are still good, it's green. Yeah, here you can see during week 5th, it's still the same. And week 6th, A had already died. There were like coagulated microalgae pieces which were dead. You can see AL is yellow. That means no microalgae. While T3 and T3L was still green. And we can see that T3L, these both are already dead. Conclude the modified and also aeration plays a huge role, including the temperature, because we saw that when the temperature was inadequate, that means less, it was not able to grow. The optimal temperature for the growth of Chlorella vulgaris is about from 25 to 30 degrees Celsius. So that was what was kept. So the conclusions are the biomass of T3 and T3L was more than A and AL. Aeration plays an important role in the growth. Temperature affects the growth of microalgae, and the purpose of circular economy was achieved. So here is the end of my presentation. I thank all of you all for your attention. Thank you.
Thank you very much. Have you any questions? Okay. Okay, thank you very much.
Thank you.
And our last lecture for today is the Crisis Leadership, It is Important, titled lecture by Andrea Fehér. The co-authors are I am Brad, Cosmina Toder, Claudia Cibulescu, and Cyprian Ruiescu, and Miroslav Reiko. Please take your presentation.
Yes, thank you. Welcome to all participants. My name is Fehér Andrea. I am from University of Life Sciences, King Mihai I from Timișoara, Faculty of Management and Rural Tourism. My presentation is entitled Crisis Leadership, Is It Important? For the beginning, I want to start with a little bit introduction what it means leadership. Leadership is a quality to influence other people to follow. So therefore, anyone who can influence people to follow them are leadership qualities. Please look at this picture, and maybe some of us find ourselves in that horse facing toward us. It is a leader. Leadership happens at all levels within organizational and societal. In fact, it's about mapping out where do you need to go to win as a team or an organization. Leadership is dynamic, exciting, and aspiring. There are several approaches. What is a leader? A leader can be a political man, a political leader, pursuing and a passionate and personal cause. Look at these personalities from the history. In the same time, leader can be a person, an explorer, striking out into the wilderness. And in the same time, we can tell that a leader is an executive developing strategy and achieve organizational goals. I try to find some definitions regarding what it means crisis and what are the elements for a crisis. I studied the literature in the field, and I found the definition related to each era, starting with 1960 to present. I find common aspects that all authors concluded that leadership crisis is a rare phenomena, but with a high impact, a high magnitude. In our research is based on a survey. A questionnaire was applied at the level of 5 large multinational corporations from Timiș County, Romania. The main objective of the research was to determine the quality of a good crisis leader. What are the key competencies of leadership in crisis situation? And what should a responsible leader do in crisis situations? So our findings regarding the first objectives is to determine the qualities of a good crisis leader, and based on the answer give the respondents, so we create the profile of a crisis leader. So the main quality are immediate adaptability and openness to change, persuasion, vision, congruence, taking responsibility, respect, and fairness in decisions. Regarding the second objective, what are the key competencies of leadership in crisis situation? The respondents give the following answers that are vision, action, communication, strength and team encouragement, and facilitating learning. Regarding the third objective, what do a good... I think that it's a problem with this slide. Okay, what should a responsible leader do in a crisis situation? So that are the follows. The first is very important to recognize that this situation is a difficult and a crisis situation. After to mobilize the company, to set clearly the priorities, to establish an action plan for achieve these priorities, to communicate efficiently. Each kind of crisis have some benefits and opportunities. So to recognize the benefits from emerging and the emerging opportunities, and to give confidence to the people in the team. The conclusions for this research are that leadership materializes from the perspective of crisis situations in the adoption of those measures aimed to adaptability. In a crisis situation, effective leaders define the challenge and vision clearly, assemble a team to coordinate and implement the change, and communicate the new vision, face the challenges during the crisis situation with courage. And another conclusion are that leaders also communicate, listen, and empathize, because team members need emotional support more than usual, and they are the ones who provide confidence and inspiration to team leaders. So that's why our research. Thank you for your attention, and good luck to everyone. Thank you.
Thank you very much, Andrea. I also want to say that for all of the speakers, that thank you very much for your participation. If I had to choose between the presentation, I couldn't choose between because I heard a very interesting and scientific presentation, and I wish you all good luck in the future and your science life and science work, and I hope you will publish your paper in our scientific journal later on. And thank you very much.
Thank you.
I would like to say thank you very much for your participation and attention. This is the end of the food processing and safety, rural and sustainable development session of the annual Wellman Conference in 2023. See you next year. Goodbye.
Goodbye.
Hi, goodbye.
Bye.